Imagine stumbling upon fragile, ancient bones. I mean, so degraded they barely hold together, almost crumbling if you touch them. How do scientists even begin to piece together a detailed story, not just a guess, from something so limited? How can just a handful of fragments actually rewrite what we thought we knew? Today, we're taking a deep dive into an extraordinary case that, well, answers precisely those questions. Our mission is to show you how cutting-edge bioanthropological techniques can transform these almost invisible remains of a Copper Age infant discovered in Faenza, Italy, into a surprisingly rich and personal story. A story about life and ancestry thousands of years ago. You'll discover how scientists figured out this infant's precise age at death, confirmed their sex using two completely different methods, and even uncovered a genetic lineage so incredibly rare it hints at previously undocumented migrations that could reshape our understanding of ancient Europe. This isn't just science, it's really a powerful testament to how even the most poorly preserved fragments of the past can speak volumes if we know how to listen really closely.
It truly is a remarkable story. And, the journey into this past began not as some dedicated research expedition, but actually as a commercial archaeological excavation. This dig was happening in Via Romagnoli, near Faenza in Italy, simply preparing the ground for, well, new construction work.
And what they unearthed there, about 1.75 meters down, was really unexpected, wasn't it?
Absolutely. The infant's remains were found inside this large hole, which the researchers interpreted as possibly an abandoned well structure. Initially, the bones were an anatomical connection, meaning, they were still positioned more or less as a body would be, oriented east-west with the head facing south.
So it suggested a deliberate burial, maybe? Even if the location itself was a bit unusual.
That's the interpretation, yes. But despite that initial connection, the skeletal elements themselves were just in an extremely poor state of preservation. This is a really common and difficult challenge with very old remains, particularly those of infants. They're just inherently far more fragile.
Right.
Most of the bones actually disintegrated during excavation, or shortly after.
Right.
They were left with only the dental crowns largely intact and a few tiny, really tiny, unidentifiable fragments of compact bone.
Wow. So with so little left, how do you even begin to place something so fragile, so degraded in time? That seems like the first hurdle.
Well, fortunately, fragments of ceramic vessels were found right there associated with the burial, including 3 distinct flask vases.
Ah, pottery. Always helpful.
Absolutely. These weren't just any pottery. Their styles, their forms allowed archaeologists to securely attribute the burial to the Italian Neolithic, that's the Copper Age, which broadly spans from approximately 3600 to 2300 BCE. They compared these ceramics to similar finds across central Italy, the Emilia area, even down to nearby necropolises. So that gave a solid time frame.
Okay, so that provided a broad window, the Copper Age.
Yeah.
But given how severely degraded the actual bones were, how much further could traditional methods, just looking at the bones, actually take them? Were they kind of at a dead end for specifics?
You're hitting on a key point there. Traditional methods, just looking at what little bone structure remained, could only provide a very broad estimate of the infant's age at death, somewhere between 1.5 and 2.5 years old.
That's a big range for an infant.
It's a huge span, when you consider developmental milestones. And this is precisely where modern bioanthropology had to step in, applying a different kind of lens.
And this is where the story gets really fascinating, isn't it? Because modern science truly shines here. The team from the University of Bologna, collaborating with, 8 other institutions, they brought a whole new toolkit to this ancient puzzle. It sounds like they almost had to invent ways to read these fragments.
They absolutely did. They strategically focused on the specific elements known to be much more resistant to time and decay: teeth and that compact bone we mentioned.
Right. Tell me more about that. Why teeth? How do they become such invaluable sources of information looking back thousands of years?
Teeth are often called biological archives for a very good reason. Their structure, their incremental growth, and their high mineralization mean they resist degradation remarkably well compared to bone. They effectively record information about growth rates, diet, even physiological stress during an individual's lifetime, sometimes with almost day-by-day precision.
Wow. Okay. And those small, unidentifiable fragments of compact bone, when everything else was basically dust, what made these seemingly insignificant pieces so useful?
It's almost counterintuitive, isn't it? These tiny pieces you can't even identify by shape, yet they often hold the biggest secrets, the genetic ones. Even small fragments of compact cortical bone can preserve enough genetic material for ancient DNA analysis when, other more informative elements have failed. They might not tell you what bone it was, but they can still whisper genetic tales.
So the combination of these resilient bits and cutting-edge tech was clearly the key. What specific techniques did they actually bring to bear on this incredibly delicate material?
The researchers employed a really powerful combination. There was dental histology, that's the microscopic study of tooth tissues. Then high spatial resolution biogeochemistry using something called laser ablation inductively. Coupled plasma mass spectrometry, it's a mouthful, LAICPMS, plus paleoproteomics, the study of ancient proteins, and of course ancient DNA or aDNA analysis.
That's quite a lineup.
It is. And it's important to note, too, that while they did try radiocarbon dating, it just didn't work here. The material was too poorly preserved.
Okay, let's unpack those. Let's start with what the teeth revealed, because that's where they got that astonishingly precise age for this infant, right?
Exactly. They meticulously selected 2 dental specimens for this really detailed study. A deciduous upper right first molar, basically, a baby tooth, and a developing permanent lower right first molar.
And from these tiny teeth, they managed to refine the age estimate from that broad 1.5 to 2.5 years down to something much, much more specific. How on earth did they do that?
By meticulously analyzing microscopic growth markers in the tooth enamel. These are tiny, almost invisible lines called daily cross striations, and slightly larger ones called retzius lines.
Like growth rings in a tree.
Exactly like that, yes.
Yeah.
Each one records a day of life or slightly longer periods. By carefully counting these, researchers could precisely refine the infant's age at death to approximately 17 months, or, about 74 weeks from birth. And this calculation even accounted for losing a bit of enamel. They added a minimum of 14 extra days for that.
That is just incredible precision from something so ancient. And you mentioned something about a birthmark in the tooth.
Yes, that's right. In the baby molar, they observed a distinct neonatal line. This is a crucial marker because it forms right around the time of birth. It allows researchers to accurately distinguish between enamel formed before birth, prenatally, and enamel formed after birth, postnatally. It showed the crown initiation for this specific tooth occurred around 184 days before birth, and its completion about 189 days after birth. So by the time the infant was born, the tooth was already about 3/4 formed.
Amazing detail. And beyond just the age, what did these tiny teeth tell us about the infant's health during that, well, tragically short life?
Well, interestingly, the absence of what are called accentuated lines. These are other microscopic markers that indicate physiological stress, like illness or malnutrition, suggest generally good health conditions. For both the mother during gestation and the infant after birth, despite obviously the infant's early death, there weren't signs of major stress recorded in the enamel itself.
It's just amazing how these microscopic details paint a bigger picture. And they even learned about growth rates compared to other ancient populations.
Indeed. For example, the mean inner enamel daily secretion rate, how fast the enamel was laid down each day for the baby molar, was about 3.13 to 3.21 micrometers per day. For the permanent molar, it was a bit slower, around 2.97 micrometers per day. What's particularly fascinating is that these rates were similar to some Italian early medieval populations, but actually lower than averages documented for, say, British Bronze Age and medieval populations.
So ancient populations might differ quite a bit from modern growth standards or even from each other.
Exactly. It highlights that variation, and it's a crucial insight for bioanthropologists trying to understand past populations, not just apply modern standards backwards.
Okay, moving on from those incredible microscopic insights, another technique they used was biogeochemistry, looking at elements in the teeth. But here, the team hit some unexpected limits, didn't they? What were they hoping to find?
Yeah, this part was a bit of a challenge. The goal with the trace element analysis, using that LAICPMS technique on the baby molar, was quite ambitious. They wanted to reconstruct dietary practices, things like breastfeeding and weaning patterns, by looking at strontium to calcium and barium to calcium ratios. They also hoped to look for environmental exposures, like lead.
So, potentially a detailed dietary history from the tooth.
That was the hope. But unfortunately, the results revealed what's called a pronounced diagenetic overprint.
Diagenetic, meaning changes after burial.
Precisely. Post-depositional alterations. Elements from the surrounding soil, like uranium and manganese, had basically infiltrated the enamel over the millennia. And these external signals completely masked the original biological signals from the infant's lifetime. It's like trying to read an ancient message that's been heavily contaminated.
Okay. So in terms of reconstructing the diet, it was essentially a negative finding. They couldn't get that data. Yet you mentioned this still matters. Why is that valuable?
Well, this kind of negative finding is actually incredibly valuable scientifically. It powerfully underscores the critical importance of assessing diagenesis, this post-mortem change, in any archaeological remains.
Yeah.
It tells researchers what they cannot reliably conclude. In this case, about the infant's diet. It prevents misinterpretations based on contaminated data.
Right, avoids jumping to wrong conclusions.
Exactly. And it demonstrates that even compromised data provides crucial information about the preservation state of the material itself. It's a vital lesson. Understand what the material can tell you, and just as importantly, what it cannot.
And speaking of limitations, you mentioned earlier that radiocarbon dating also hit a similar wall.
That's right. They attempted radiocarbon dating on about 250 milligrams of the bone fragments, but it was unsuccessful. The collagen yield, the protein needed for dating, was extremely low, less than 0.1%. This indicated insufficient preservation and, again, extensive diagenesis. It just wasn't possible to get a reliable date using that method.
So the pottery confirmed the Copper Age context, but no precise C14 date for the burial itself.
Correct.
Okay, so despite those challenges with dating and diet, other major breakthroughs were made, particularly around identity and, well, this really uncommon ancestry. Let's start with how they confirmed the infant's sex, not just once, but twice.
Yes, they got a double confirmation, which is always reassuring. First, paleoproteomics, the analysis of ancient proteins, was done on a small enamel fragment. It yielded very clear results. They identified peptides associated with both the AMEL-X gene, found on the X chromosome, and the AMEL-X gene, found only on the Y chromosome. And, crucially, there was a strong signal from the AMEL-specific peptide. This unambiguously confirmed the infant's sex as male.
Okay, protein analysis says male, and the second confirmation came from the genetic.
Precisely. Ancient DNA analysis from one of those unidentified bone fragments further corroborated this. Even though the amount of the infant's own nuclear DNA recovered was very low, the ratio of sequences mapping to the X chromosome was consistent with a male assignment.
So two independent lines of high-tech evidence pointing to the same conclusion, a male infant.
Correct. Very solid finding.
Now, for what I found to be the really intriguing part, this matrilineal mystery, despite the low quantity of nuclear DNA overall, they were able to reconstruct the infant's mitochondrial genome almost completely. And this revealed something highly unusual, didn't it?
It absolutely did. This was perhaps the most surprising finding. The analysis revealed an uncommon mitochondrial haplogroup, V plus at 72.
V plus at 72. And what makes that particular haplogroup so rare or significant?
Well, what makes it so rare is that this specific genetic marker, V plus at 72, has only been identified once before in any ancient samples published so far.
Only once? Where was that?
In an Neolithic necropolis in northwestern Sardinia. And those remains dated to between about 2454 and 2001 Cal BCE. So roughly contemporary, but geographically quite distant.
Sardinia, Italy. And in modern populations today, is it common anywhere?
Almost nowhere in Europe. In modern European populations, it's virtually absent. The exceptions are higher frequencies found among the Sarami people, way up in northern Fenno-Scandinavia.
Wow, okay.
And some scattered groups along Spain's Cantabrian coast, but otherwise extremely rare, which makes finding it in Copper Age Faenza really significant.
So what does this single rare genetic marker found in this one infant from Copper Age Italy, what does it actually mean for our broader understanding of ancient Europe? Could this one piece of DNA really suggest, well, bigger movements?
It strongly hints at it, yes. It suggests previously undocumented prehistoric migrations or perhaps long-distance cultural interactions across Europe thousands of years ago. It implies a far more complex tapestry of ancient movements than our current models might assume. It challenges us to think about how connected these early European populations might have been.
So it paints a picture of a past that was maybe far more dynamic, more interconnected than we often picture.
That's certainly the implication, yes. A past with perhaps unexpected links across vast distances.
And I imagine this opens up a whole new avenue for research, doesn't it? Pushing scientists to look for more evidence of these connections.
Absolutely. It's really important to stress, though, that because the nuclear DNA coverage was so low, we can't make broader genome-wide inferences about this individual's overall ancestry just yet. This really underscores the critical need for more paleogenetic data, specifically from Neolithic communities in Adriatic Italy. We need more samples to see if this was an isolated case or part of a larger, currently unknown pattern of movement or connection.
Fascinating. This whole deep dive really illustrates the immense power of integrating all these different advanced techniques, doesn't it? Even when traditional methods hit a wall, like with the bones being so degraded, and even when individual analyses face limits like the contamination affecting the diet study or the lack of collagen for C14 dating, when you combine all these methods, their combined application can yield profound, even really surprising insights. It's like each piece of the puzzle, even a broken one, adds something vital to the bigger picture.
Precisely. It's a perfect example. The study of this Copper Age infant from Faenza is a compelling case for what researchers call osteobiographical objectives. It's about moving beyond just broad population-level generalizations and really trying to meticulously reconstruct individual life histories as much as the evidence allows. It shows us that every fragment, no matter how small or how degraded, potentially holds a unique story, a story just waiting patiently to be told if we have the right tools and the persistence to look.
So what does this all mean for you, listening?
Well, I think it's a powerful reminder that history isn't just about the grand narratives, the kings and empires. It's also found in these incredibly intimate details of individual lives. Even the life of a 17-month-old infant from thousands of years ago, waiting patiently to be revealed by dedicated scientific inquiry. It makes you wonder, doesn't it? What other seemingly insignificant pieces of the past might still be holding extraordinary secrets about ancient journeys, maybe forgotten connections, or just the quiet, everyday lives of our distant ancestors, all just waiting for the right tools, the right questions to unlock them.